Okay, today we are continuing chapter 1. We are doing the second half of section 4, which is the last section in chapter 1 that we'll be covering. Okay, here are our goals for the day. Summarize the classroom lab safety rules, compare light microscopes and electron microscopes, identify the function of each of the major parts of a compound light microscope, differentiate between magnification and resolution, and determine the magnification of a microscope. Okay, just a note, I want to make sure that you guys remember the lab safety rules and that you can identify these lab safety symbols. You should be able to identify each symbol and summarize them or provide an example for each. And I will ask you to do this on a quiz in the future. Okay, so today we're looking at light microscopes and electron microscopes. These are the two main types of microscopes that we use in biology. So the light microscope, or more specifically the compound light microscope, uses light to kind of view an image. So the light passes through the specimen, and lenses in this area and this area magnify that image so that we kind of get an up-close view of the cells. So for example, these are plant cells, and you can see the chloroplasts in them. This is what an image would look like in a light microscope. And it's important to note that the light has to be able to pass through the specimen. So the specimen must be very thin. So typically we will get very thin slices that are placed onto slides so that the light can go through it and we can view the image clearly. Okay, so the parts of a light microscope. The first part is the eyepiece. So this is where you put your eye. It's also called the ocular lens, and it magnifies the image 10 times. There are also what we call objective lenses, or objectives. These further magnify the image. There are different powered objectives, so the lower power or higher power, and those allow you to kind of choose which magnification you want to have to view your specimen. Okay, then we have the coarse adjustment and the fine adjustment. The coarse adjustment moves the stage up and down in large increments, and the fine adjustment moves it in small increments. So typically you'll kind of get in the general area you want with the coarse adjustment, and then you'll use the fine adjustment to really zero in specifically and get that focus that you want. And it is also important to note that when using high-powered objectives, which are the long ones like this, you only use fine adjustment because if you use coarse adjustment, the lens is so close to the stage that the stage could ram up into the lens and damage it. So we only use fine adjustment so that the stage is only moving in small amounts because we do not want to damage these microscopes as they're very expensive. Okay, next we have the stage, which is this platform here, and the stage clips, which hold the slide into place. And when we store the microscope, we always have the stage lowered and the lowest power objective or the shortest objective chosen. And this is because, again, we want to make sure that the stage is not anywhere near the objective lenses so that they do not get damaged. Okay, the next two parts are the diaphragm and the light source. The diaphragm typically lets you control how much light gets through. However, our microscopes do not have a diaphragm. We do have a light source, and that is where the light comes from that passes through the specimen. Okay, the next two parts are the arm and the base. So these are the support system for the microscope. The arm is here and the base is here. And it's very important that whenever you carry a microscope, you have one hand on the base, one hand on the arm. Carry it with two hands to make sure that it doesn't get dropped and that it is being carried properly and supported properly. Okay, homework question number 1. In the following image, identify the stage. Which letter represents the stage? Homework question number 2. In the following image, identify the ocular lens. Which letter represents the ocular lens or eyepiece? Homework question number 3. In the following image, identify the arm. Which letter indicates the arm? Homework question number 4. In the following image, identify the objectives. Which letter represents the objectives or objective lenses? Homework question number 5. In the following image, identify the light source. Homework question number 6. In the following image, identify the fine adjustment. Okay, we have a couple of vocab terms associated with microscopes. The first is magnification. So this has to do with the increase in size of an image using lenses or mirrors. So if we have a smaller image, we can magnify it to make it bigger. And resolution has to do with how clearly you can see that image. So instead of going from something smaller to larger, now we're going from something blurry to something with finer detail. Okay, with light microscopes, the way you can determine magnification. So the first thing to note is that the ocular lens is typically times 10. So that's the part where you put your eye. And then the objective lenses, which you see here, the red is times 4, the yellow is times 10, and the blue is times 40. So the way you determine the total magnification of what you're viewing is by multiplying the ocular lens times the objective lens. So if we're using the red objective, we'll have 10 times 4, which is 40. If we're using the yellow objective, 10 times 10 is 100. Or if we're using the blue objective, 10 times 40 is 400. So homework question number 7. On a microscope, the ocular lens has a magnification of 10 times, and the chosen objective lens, which is blue, has a magnification of 40 times. What is the total magnification this microscope is currently producing? Okay, and homework question number 8. I want to view a slide at a magnification of 100 times. Which objective lens should I use? A, the red objective lens at 4 times magnification. B, the yellow objective lens at 10 times the magnification. C, the blue objective lens at 40 times magnification. Or D, the ocular lens at 10 times magnification. Okay, so we looked at light microscopes. Now quickly we're going to look at scanning electron microscopes. So these are very similar to light microscopes, except they are a lot larger, a lot more expensive, a lot more technologically advanced, and instead of using light, they use electrons. This is an example of the type of image you will see with a scanning electron microscope. And you can see that there's a lot of detail, very fine detail. And again, this is a plant cell, and these are the chloroplasts. Some things to note about the electron microscope is they do have more powerful magnification and resolution. However, you can't use them to view living things because the specimens have to be placed in a vacuum chamber in order to do them. And artificial color is added to the photos afterwards. The